Episode 5. Can't even believe it. Plant a seed and just give it some sun and water and hope it grows. It wasn't meant to be a pun, but it kind of is. Welcome to Absolute Genius, a new podcast series from Thermo Scientific. I'm Jordan Ruggieri.
And I'm Cassie McCreary, and for today's episode, we're thrilled to speak with Dr. Ray Ketchum about his work.
Ray is the group leader for manufacturing sciences and technologies at Gene-ius, a company focused on modern agriculture sustainability. He holds a PhD in botany and boasts over 30 years experience working in plant science. He also gives some great career advice on the episode today that I found extremely useful. We hope you enjoy our conversation.
101 plant puns. I beg your garden. That one's mine.
All right, I'm going to say, have you bought any plants lately, Cassie?
What we do here is we produce microbial consortia to use in agriculture. So these are basically microbiomes that we manufacture, we sell to growers, and those are applied to the field. What I do is I lead the group that does much of at this point, it's the research and development, as well as the technical production for the bacteria that we grow, and also for doing the analysis of those bacteria. My group kind of coordinates the quality control tests that our quality manager and our quality technicians actually use.
That's awesome. So you talked about consortia. Can you elaborate a little bit on what that means and how that's important?
So Gene-ius manufactures three main agricultural products at this time. We have others that are in development, but those are our commercial products. One of them is a microbial consortium, which contains 22 different bacteria. These are all soil bacteria. They're nothing genetically modified. All of our products are organic certified and OMRI certified. So these are just naturally occurring soil bacteria that we've grown them up separately, at least to isolate them. But then as we manufacture them, we group them together. And so we grow up this large fermentation, which contains a collection of anaerobes in one fermentation and aerobes in another fermentation. And we blend those together. And these are all bacteria which are intended to improve plant health, mostly by increasing the availability of nutrients in the soil. Some of the bacteria actually fix nitrogen. Some of them help to solubilize inorganic nutrients that are in the soil, like phosphorus or certain types of inorganic potassium, and make those available to the plant. So all these bacteria have been isolated essentially from the rhizosphere, which is the part of the earth that surround the roots of each individual plant. If you pull up a plant and you've got that big bundle of dirt, there are a lot of bacteria that are in that soil that are very closely associated with the roots. And there's sort of this beneficial interaction between those bacteria and the plants. Plants actually provide the bacteria with certain nutrients, and in exchange, the bacteria also help to solubilize some of the materials or actually produce nitrogen that get absorbed by the root. So there's this interaction. And we're trying to exploit that by making up these groups of bacteria that you can actually add to the soil. So the consortium refers to just that large collection of bacteria in a single sample or a single product. Now, I mentioned also that there are two other products that we manufacture. So we've kind of taken this and we've gone one more step. And what we've done is we have a production facility in Mexico that uses these same bacteria to help break down shrimp biowaste in Sonora, Mexico, along the Gulf or the Sea of California, the Sea of Cortez. There's a lot of shrimp farming that's done down there. And, after the shrimp are deveined and the shells are removed and they're basically cleaned up, there's all this waste material that's left over. And so we take that waste material and we ferment that with these same bacteria. And from that, we produce two additional products. One is a biostimulant product, which is the liquid that's left over from that fermentation. And the other is all the solids that are left over are dried and milled down. And that's our third product. So, we've been trying to be this green company where we're helping to reduce waste by actually using that waste in different ways and applying that to agriculture. And it's actually worked quite well.
Question about the shrimp. I'd love to talk about that. How did you even find out that that shrimp waste can be something that's impactful for plants and for the soil?
Shrimp and almost any other type of shellfish or that type of waste, if they contain protein, they contain a source of nitrogen. And nitrogen tends to be the nutrient that plants need the most. They can't make it themselves. It's returned to the atmosphere fairly readily. So any source of nitrogen is usually a good thing. The other thing that shrimp and also other types of shellfish have, crustaceans in general, have chitin in their exoskeletons. And that chitin can also serve as another source of kind of a more slowly degrading nitrogen. As long as you have bacteria that are in the soil that are able to break that down, they can break down that chitin into to nitrogen as well as some other carbohydrates. And again, those are all very beneficial for plants.
So it was looking at the the chitin in the material that the shellfish have and seeing how you could apply that into the consortia and what you offer to try and get more nitrogen into the plants is kind of how that stemmed. Is that a correct assumption?
Right. I mean, that's basically the end product works that way. So that the solids that are left over, that's the material that contains the chitin. The other interesting thing is when you actually do this process, you separate the solids and the liquids. The liquid portion of this gets, the lipids actually get removed from that. And the lipid or the material that's left over is this liquid portion that's in many cases kind of what's left over from the bacteria when they're actually using this as a food source. So it's some of the metabolic products that the bacteria produce enriched in this liquid. What's fascinating about that particular liquid is it has it's kind of like a stress relief mechanism. It's a way of helping plants to recover from certain types of stress, like water deficit or in some cases herbicide or pesticides, drift or anything like that. And it seems to be very effective in terms of relieving stress.
It's like a seafood broth for when you're sick. A nice broth to calm the throat. It does the same thing for the plants.
Exactly. Instead of a chicken broth, it's more like a shrimp broth.
Some might call it Shrimp-ly amazing.
Oh.
I'll see myself out. You two have fun. Bye now.
They might call it that, yes.
See you next time on Absolute Genius. Yeah. Carry on.
So talk a little bit about the R&D behind some of these products. Do you look at individual strains or species of bacteria and kind of try to figure out how they might interact as a consortium, and if it's worthwhile to add them in, or how does that R&D kind of function?
We have a large consortium or a large collection, I should say, of bacteria that have been collected through the years from a lot of these field sites and things that are, sort of in our stores in our master cell bank. And that gives us a large library to go back to and pick out certain types of function. A lot of the bacteria have been screened for metabolic functions and characterizations. That's one of the things that the R&D group and my group does.
Okay.
We screen different bacteria for different abilities in terms of breaking down waste or chitin, for instance, nitrogen fixation, solubilization. But then we also will take individual strains. We will DNA sequence them. So those are some of the types of activities that we do. But probably the main thing that my group does and spends most of their time on is just figuring out how to grow these bacteria together. What's a little bit unusual about our company compared to other companies is a lot of companies will put a bacterial product together that may have 4 or 5 bacteria in it. in which case they've grown each of those individual bacteria separately. One of the things that we found, and it kind of applies to this biostimulant product that I was telling you about, this liquid portion, is that there is a benefit to growing all of the bacteria together. There seems to be communication between certain bacterial strains, kind of almost a interaction between those strains. And if you think of kind of some of those ecosystem studies that you might have done in biology where, you had a fish tank and you threw some, duckweed in there and maybe some algae and you threw some fertilizer in there and then you just kept taking samples throughout the year and you saw how that thing changed over time. That's kind of what you see with some of these bacteria as well, is the whole population will change at certain times during the fermentation. And we want to get to a point where things are pretty much stabilized. We know the numbers of certain types of bacteria within there, and we know that's going to be an effective product once it's applied to the field.
Interesting. So you're seeing a benefit to growing them all together. So there's not like some kind of like competition in a sense between the bacteria, or?
There is.
Okay.
And again, there absolutely is. And that's where the real fun starts, is figuring out how do you get these things to grow together so that they'll all come up, they'll all reach a certain titer, a certain population within that product. There's competition, and, if you don't do it right, there will be certain bacteria which will completely dominate and mess up the whole culture. That's sort of the proprietary work that we have in terms of figuring out how do we get them all to grow together and how do we get them all to get up to that particular titer. That's where the magic happens.
That's very cool. Now, I would assume as you kind of scale from R&D to more, production level for a product, there is some difficulties that arise in the scale up. Is there a way that you need a QC or QA as you kind of scale up the product and make sure that you're seeing the same kind of breakdown of bacteria?
That is one of the main challenges is making sure that we're hitting a certain titer. For our main product, we have a specification on our label which we have to meet just from a regulatory standpoint. QC is very much involved in that with us. The other thing that we need to do too is, depending on if it's a new product, we need to figure out, once we're able to grow things together, well, what is that titer that we can reliably hit? So there's a lot of work done on seeing, if we adjust this parameter, can we get everybody up here, or, do we have to settle with everybody down here, or what? So to get those titers, if we're working with individual bacteria, it's super easy to do. You just plate them out. But if you have a collection of bacteria, that's where things get a little bit more complicated. And that's where the digital PCR comes in.
How does digital PCR as a technology play a role in that process?
We wouldn't be able to do what we're doing without digital PCR. The reason for that is because we have a product that has 22 different bacteria together. It's impossible to plate these bacteria and count them, all at once. Some bacteria don't plate very well. Some of them are anaerobes, some of them are aerobes, so they can't grow under the same conditions. So all of these variables make digital PCR really a keystone to the technology and to our quality control. So what we do is we do a whole genome sequence for each of our bacteria. We figure out within that genome which genes are single copy and which ones are unique to that particular species. And then we design primers and probes for that portion of that particular gene. Or in some cases, it's just a DNA sequence. It may not actually be a coding gene. And then once we have that, we can take a very complex mixture of bacteria, do a total DNA extract on that, and then analyze that, again, with the assumption being that every signal that we get on digital PCR relates back to one single bacterium. And by that, we can actually get some pretty good numbers.
Makes sense. Do you run into any inhibition problems as well when it comes to bacteria or some of these samples? PCR inhibitors or any type of inhibitors that might impact, say, real-time PCR versus digital PCR?
I would say that, at this point, we haven't actually done the real-time PCR versus the digital PCR comparison to see if there's a difference there. I don't suspect that there is, simply because of the way the DNA is extracted. Most of those inhibitors should be removed. But there is another issue that we have run into recently, and I think it's something that the industry as a whole is starting to notice a little bit more, and that is that, when you take this complex collection of bacteria and you're doing a consortium on them, whether it's from a human gut microbiome, the soil microbiome, or whatever, there has been this assumption that your DNA extraction is extracting the DNA from all of the bacteria equally well. And that's simply not the case. In our case, what we found is that a lot of our bacteria are spore forming. And if they've formed spores, we just have a heck of a time really getting the DNA out representative of what's actually in the product. It turns out that we've been way undercounting what our product actually has. So those are some of the issues that we've run into. And we're working on those. So we don't have a solution to those yet. We're really not sure exactly how much of an impact this is going to have. We started working with some of those 22 species, but we haven't done all 22 species yet. So this is something that we're working on right now.
How does this product impact the customer? Is there an impact on food production per se for farmers?
So what our product does is it improves the yield in the field. We've seen individual studies where, depending on the crop, it could be up to a 30% increase.
Oh, wow.
That's really unusual. More frequently, it's in the 5 to 15% range. But what the product does is, as I mentioned, it does fix nitrogen. So it adds additional nitrogen to the soil. It solubilizes any sort of inorganic nutrients that may be in the soil already. Some of the bacteria help to break down residual field waste, a lot of the straw left over from, and canes that are left over from harvest, they help to break that down. So there's a lot of benefits that way. As I mentioned also, the other two products, one is a stress relief product. That's our Be Sure is what it's called. Invigorate is the consortium that we're talking about. And Uplift is the chitin-containing product as well.
Okay.
So all of those, yes, increase in crop yield. And mostly it's for agricultural crops.
Okay.
Some of the Be Sure has been used very successfully in turf. So
any studies into potential impacts on, say, climate change?
We haven't done any studies specifically about these bacteria. Certainly, one of the things that they will provide is additional nutrients to the plants without additional artificial inputs, so chemical fertilizers. So at least in that regard, that 5% increase in yield is coming not at the expense of using additional petrochemical-based products and gas and diesel that's used in the field.
Okay.
But just from the bacteria themselves. But we haven't really looked at any sort of carbon sequestration issues with the products yet.
But maybe some potential like chemical runoff or things like that. Interesting.
Right.
Okay.
Right.
Taking a quick break from our conversation to tell you about Applied Biosystems Quant Studio Absolute Q dPCR system. This instrument enables quantification of your targets without the need for standard curves in only 90 minutes. Digital PCR can be as simple as preparing your samples, loading onto the plate, and running the instrument.
Unlike other digital PCR systems, the Absolute Q dPCR instrument does not use emulsion or other droplet-based methods to compartmentalize reactions. In fact, the microfluidic array plate technology enables consistent delivery of more than 20,000 microchambers.
And Thermo Fisher Scientific has a suite of dPCR assays for applications like AAV viral titer quantification, liquid biopsy analysis, and wastewater surveillance. You can learn more at www.thermofisher.com/absoluteq or visit the Absolute Genius webpage. Again, that's www.thermofisher.com/absoluteq or visit the Absolute Genius webpage.
The Applied Biosystems Quant Studio Absolute Q dPCR system is for research use only, not for use in diagnostic procedures. Let's get back to our conversation.
I would be very excited to welcome Ray to Cassie's Career Corner. Step into my office.
You've got the alliteration down, so you have to.
I know, how lucky was that? Thanks, Mom and Dad, for naming me Cassandra, the way you did. Jordan, you can come in too. Come on in. Here we go, everybody.
Oh, of
We're all
course.
here in the career corner. This is the part of the podcast where we talk about obviously your career, maybe some fun stories and things along those lines. So we're gonna run the gamut here a little bit. Correct me if I'm wrong, but I believe your educational background, you have your undergraduate work was biochemistry and botany, and then your PhD was plant biology and botany. Do I have that correct?
Correct. Yes. My undergraduate background was at Connecticut College, a small liberal arts college. And that's where I first got introduced by Professor Scott Warren, who is a good friend still. He introduced me to working with plant cell cultures as part of a plant physiology class. And I ended up actually taking over the cultures that he had been keeping going just as part of a work study project. And that was, I don't know, there was just something about, keeping plant cells going sort of indefinitely in sterile conditions that just fascinated me. And so that's kind of where I got involved in the whole sterile technique thing. At the time, I was working on plant salt tolerance in plant cell cultures. And the reason for that is because Scott had actually started these salt marsh grass plant cell cultures that were resistant to sodium chloride that you added to the medium. And that also really fascinated me. So I kept working on those, and eventually we got a paper out of it. That was something that got me involved in graduate school. I wanted to continue working on salt stress tolerance in plants and salt stress mechanisms. I've been working on those plant cell cultures all this time, and that's kind of the work that I was doing with my graduate work.
Very cool.
And all of that, especially the stuff that was working with the suspension cultures, and so that was the other things. Almost everything I worked with were these liquid cultures. So it wasn't necessarily just callus on agar. I was doing a lot of work with plant cells in liquid culture, and that's sort of intrigued the USDA because they were working on plant cell cultures of Taxus brevifolia, which was the Pacific yew, and looking at it for as a source, in cell culture of the anti-cancer drug Taxol, which at that time was being harvested in forests, for the bark and for that anti-cancer drug. And some of the people I worked with, Donna Gibson, and the work that was first started by Alice Kristen, was looking at using plant cell cultures as an alternative source of supplying that particular anti-cancer drug, since the trees themselves are small, they're kind of scraggly looking, they take forever to grow, they're very slow growing. They are. I've been out in the forest. So I'm in the Northwest right now, and I've been out in the forest, and I've found them out there, and they're just not attractive trees most of the time.
That's still amazing research to be a
It
part
is.
of. That's
It was. Yeah, I was really fortunate in being able to kind of participate with that. And that has pretty much been most of my career, working with USDA to get those cell cultures going, for them to do research on, for all of us to do research on. As a visiting professor at Cornell University, continuing work with that. And then as a postdoc and eventually a research professor in Rod Crotto's lab at Washington State University, because he was the one who was doing most of the molecular biology of this pathway in the plant. And it was just a really nice collaboration for me because, I had these plant cell cultures that were producing this anti-cancer drug, and he had this stable of graduates and postdocs who were doing a lot of molecular biology to try and pick out all of those genes in that pathway, and this was a really nice source that they could use to do that. And I was very fortunate in being able to kind of hone my molecular biology skills in that lab as well while I was working with them. So.
It's an amazing way to visualize a group of students. A stable of students.
That's right.
There was a lot. We had there were over 30 people in the lab at one point, so it was quite an operation. So.
And you've shifted since, well, kind of from the plant cell culture to now this sort of the soil microbial focus and everything. So What was that like making that kind of that change? And then what was sort of the drive there for you?
Well, yeah, it's sort of a continuing story, right? All of these things are connected. It's all about the networking. The cell cultures that I started while I was at Cornell University were used by a graduate student colleague of mine who was at Cornell after I'd already left. And so I had left Cornell and gone to Washington State. And he was working in Professor Mike Schuler's lab at Cornell in the chemical engineering department, and was actually working with these plant cell cultures that I had developed. So we knew of each other kind of through Donna Gibson, my former USDA boss, but we hadn't really met each other. Anyway, Harry ended up somehow coming back to Portland and working with a company that was also doing some of the work with these plant cell cultures, these Taxus U plant cell cultures. So he knew of my work that way. And we had this small company, biotech company called Diana Plant Sciences in Portland. And one of the things that we did is we actually produced essentially what we called cosmeceuticals. So these were essentially just freeze-dried plant cells that were very high in antioxidant content. And those were incorporated into French cosmetics. This was sort of my first exposure to actually growing plant cell cultures in larger fermenters. So that was my work with Harry. Harry eventually left Diana Plant Sciences and then joined Agrinos. And at that time, Agrinos was building this large fermentation site where I am sitting right now in Clackamas, Oregon, which is just outside of Portland. And Harry eventually called me and said, hey, I need somebody to run the manufacturing science and technology group to help go from R&D to scale up these bacterial cultures into these larger fermenters, which is essentially what I was doing at Diana Plant Sciences, where we're going from, plant cell cultures on plates to smaller shake flasks to 250-liter fermenters. So that's how, like a long, long story as to how we went from plant cell cultures into microbes.
Oh, I love that. That's great. And I think you hit on, without even me prompting you, so well done you, snaps hooray. You hit on, I think, something really important, and I think it's an important piece of actually career advice, is it's all about the networking. And I think that a lot of people would find that that's a pretty important thing when it comes to the trajectory that their career will take. Do you have other pieces of advice? Like if you could go back and tell yourself when you were a graduate student, or to somebody who is brand new and just entering, either the industry or even academia, what's a piece of advice you might give?
Yeah, I mean, the networking thing, it's funny because I always sort of poo-pooed it and just said, it's really not about the networking. It's about your abilities and, what you do, and which is nonsense. That's sort of the old Ray coming back and telling young Ray, don't be such an arrogant jerk. It's funny because I had a colleague when I was with USDA who mentioned the fact that he'd never gotten a job that he applied for, or he'd never applied for a job, yet he was completely, fully employed, and at that time was, a researcher with USDA. And, for him, it was just, like he said, it was, or like we discussed, it was all about networking. It was about, somebody knowing somebody who had a job opening up, who recommended you for that. And, to some extent, almost every job that I've had has been exactly that way. For 30 years, it's really been connections. So you really can't, I don't think, undersell that, or, you certainly shouldn't underestimate the value of that. As far as career advice goes, and just kind of being arrogant, when I was applying for grad schools, I was applying to programs, I'd read papers by a lot of these different researchers, and found a couple of positions or a couple of labs that I really wanted to work in. And so I wrote directly to the professors, in those labs. I made my decision based on whoever was going to pay me the biggest stipend, which I'm sure I'm probably not the only person who's done
I
that.
sincerely doubt it, yeah.
Yeah. But where the arrogance came in is I said, I didn't want a teaching assistantship. I wanted somebody who was going to give me a research assistantship, because research was the thing that was really interesting to me. And so I ended up going to Colorado State only to find out that after I'd been there for, on my research assistantship, that expired after like 2 years. And then I had to go for a teaching, I had to get a teaching assistantship. So I had to do the teaching anyway. And I loved it. I absolutely loved it. I had a great time teaching. And if I'd known it was going to be, that much fun, I probably would have done that initially. That's one piece of advice is, listen to the people around you, especially your professors. Don't assume that you know more than they do, because you don't. And if they say, this is a good idea, you should actually go there, really listen to that advice. It's good advice.
I wouldn't consider myself super crazy far along in my career, but even I would look back and say, well, I might do this different or I might do that different. And I think it's really easy at any point in your career to kind of pause and take a second and say, well, if I had done that, what would it look like today maybe, or something like that too. And so I think it's just kind of, it's interesting from your perspective now and seeing how, and then ultimately something that you were very much not interested in, you really ended up enjoying in the end. So
you bring up an interesting point that I didn't touch on. So, I said that my undergraduate was at Connecticut College where, I double majored in botany and biochemistry. I didn't start there. I started at Cornell. I started at Cornell in mechanical engineering, and it was a disaster. I hated it. They weren't very fond of me either. So, when I finally left, I swore I would never go back there. I just had such bad feelings about the place. And so when I applied for my first postdoc at USDA with the Taxol project, Donna Gibson was at the Southern Regional Research Center, which was in New Orleans. And I interviewed down in New Orleans with her. She offered me the job. And just like before I was getting ready to pack up and move down there, she called me the week before and said, Ray, my husband just took a job up in Sayre, Pennsylvania, and I'm going to be working at the Federal Nutrition Lab at Cornell. Do you still want the job? And I just hung up and I said, I swore I would never go back there. And so I ended up back at Cornell. And that actually, that experience was much better than the original experience as an undergrad. Mechanical engineering was not going to be for me, and I swore I'd never go back to Cornell. But both of those things, well, not both of those things, but going back to Cornell definitely did happen. So,
yeah, we'll just consider the mechanical engineering years just like a wash. It's fine. I have it too. Yeah, I started down the path of I was interested in being a forensic toxicologist, and then I started down that path, and I was like, well, I hate this, so and here now I'm a digital marketing manager, so go figure. But
so speaking of small worlds, when Diana Plant Sciences closed, they were actually the Diana Group was bought by a chemical company called Simrise, and they closed us in Portland. And so there was a year there where I was between jobs, and I became the scientific director of a forensic toxicology lab.
Oh, weird.
And that was all of the analytical experience I had as a postdoc, which was, the gas chromatography, HPLC, LC mass spec, GC mass spec, all of this analytical experience I picked up at Washington State University. Was just, oh yeah, now let's analyze urine. So it's like,
that's exactly right. That's
sure.
Exactly right.
And that's exactly why you got out of it, right?
I mean, you're not wrong. So, right. Wow, what a weird set of connections.
Well, if you get to be as old as I am, you're going to connect with probably, somebody at some point. It's just inevitable. There aren't that many other people. So.
Oh, that's so funny. That was Ray Ketchum, group leader at Agrinos in Portland, Oregon. Thank you so much for joining us for today's episode of Absolute Genius. Stay curious, and we'll see you next time. If I try any more plant puns, I might soil myself.
That's a good tie back to Theses Happens.
We love a theme.